So we're picking up on part 2 where our video cut off. Sorry about that. And so we're going into microbial growth. We're picking up with the minimum inhibitory concentration test. Now with this minimum bacterial concentration, sometimes what it's referred to, or MBC test, this is going to determine the amount of drug that's required to kill a microbe. So we're going to have samples that are going to be taken from that Mick broth dilution test, and we're going to transfer that onto plates. Those plates will contain drug-free mediums, and we're going to monitor it for bacterial replication to see if it actually killed it or if that bacteria can still grow. The lowest concentration of drug where no growth is going to occur is going to equal that MBC, or minimum bacterial concentration. So that's going to be important. So that's what you would do with a minimum concentration test. So that's going to be on this one. So we take our sample, we put it there, no colonies grow, and we continue, we dilute it more, no colonies grow. We're trying to figure out the least amount of drug that we have to use because we don't want to use too much if we can use much smaller amounts. Now, different routes of administration. External infections may require things that just go on the outside. So you might have topical or local administration to be most effective for these types. You can have internal infections. That's where you might have some oral infections. So you might have some self-administered medications. They may require no needles. You could have an intramuscular medication that needs to go in that's given directly through a needle into a muscle. So it goes directly into that muscle in order to deliver the medication. You might have an intravenous delivery, and so that's delivered directly from a needle into the bloodstream. So then it makes its way around the body that way. Now, safety and side effects. We're going to have 3 main categories here. First, we have toxicity. That's going to be the cause of many adverse reactions that are poorly understood. So drugs may be toxic to certain organs, and consideration is going to be needed when prescribing these drugs, especially to people that might be pregnant, like pregnant women, and they could have adverse effects on the fetus. Allergies. These are relatively rare. Some may be life-threatening. It could go into anaphylactic shock, or you might have what's known as, sometimes you have red man syndrome, where it makes the body release a lot of histamine and you look bright red. It almost feels like you have a sunburn. The skin can be really hot. It could create things like hives, or it might cause you to become sick on your stomach. It could cause diarrhea, or it could cause vomiting. Disruption of normal microbiota, that is going to be the third one. And so that may result in a secondary infection. So let's say that you killed the good bacteria in your stomach if you have some bad bacteria there, or let's say if you kill the bacteria on your skin, you still are going to have fungal colonies that are there. So then they can come in and they can overgrow and take over that area, and they could actually cause an infection on the skin from a different type of organism or a different species of microbe that you killed off because it no longer has competition. So you could have overgrowth of that normal flora that causes super infections. So now you've got more than one infection going on. The greatest concern is going to be for patients that are in the hospital, because being in the hospital, you're going to have lots of different types of infections there. So you're more likely to get a infection in that area. So with this, looking here, we see that black hairy tongue. This could be from different types of antibiotics, such as penicillin, amoxicillin, things like that. So what's happening is it is causing the normal flora that is normally there. It no longer has competition, so the other bacteria produce waste products that then can cause it to look hairy and cause the filiform on the tongue to get this look to it. So development of resistance populations. That is something that we worry about with antimicrobial drug resistance. Some populations may contain organisms either partially or completely resistant. 2 ways for resistance to occur. You could have a new mutation of a chromosomal gene, so that genes can change and mutate and make themselves so they are not susceptible to the medication anymore, and so they're able to live even when the medication is administered. or acquiring a resistant gene from R plasmids. So R plasmids are going to be extra chromosomal pieces of DNA. They can be acquired via horizontal gene transfer. So it's transformation, transduction, and conjugation. So basically what happens is the two microorganisms line up close to each other and they are able to swap some of their DNA or some of their genes. And when they do this, they can send over some genes that made them resistant to that drug. And when they send it to a microorganism that's not resistant to it, then it becomes resistant. So resistant individuals do not experience exponential growth until non-resistant individuals are going to be inhibited or die. Results are going to be from less competition between these individuals. So they basically have a free-for-all because they're not competing against the other microorganisms. So with our mechanisms of resistance here, we have 7 of those. 1, producing enzymes that deactivate the drug. So that's one way it could do it. The second way, slow or prevent entry of that drug into the cell so it can't get in to kill the microbe. Third, it could alter target of drugs so it binds less effectively to the cells. Fourth, it could alter metabolic chemistry and pathways. Fifth, it could pump antimicrobial drugs out of the cell before it can act. Sixth, development of biofilms, which are going to retard drug diffusions and slow metabolic rate. And seventh, it protects targets of an antimicrobial drug to inhibit its binding or to inhibit those proteins that help it to enter. So we can have multiple and cross resistance. If it's multiple drug resistant pathogens, it's going to be often known as a superbug. I'm sure you've heard that in the news or you've heard people talk about that. These pathogens are going to be those that are resistant to 3 or more different types of antimicrobial agents. We have also what's called cross resistance. That's a resistance to one antimicrobial agent, and it confers to the resistance to other similar drugs as well. So in order to, or with retarding resistance, there's going to be 4 ways that we have retardative resistance. The first one, maintain high concentration of drugs in a patient for sufficient times. This is going to kill all sensitive cells and is going to inhibit others so the immune system can destroy it. Secondly, the use of antimicrobial agents in combination. So it promotes synergism, which results in the efficacy that exceeds the efficacy of one drug alone. So it makes it more efficient to have 2 that come in and work together. Third, use antimicrobials only when necessary. And then fourth, develop new variations of existence of drugs and new antimicrobials. So that's going to finish us up for this unit. I will see you back for Unit 7.